Greetings to all participants. Welcome to the pre-event IBASC 2021 organized jointly by Chemical Engineering Universitas Gadjah Mada and International Bioprocessing Association. We would like to extend a very warm welcome to all of you. For all the participants, do not forget to fill out the attendance list in the form as it is one of the terms to receive the e-certificate. You can scan the QR code or access the form through this link.
Good afternoon, ladies and gentlemen. Dear Dr. Yuni Kusumastuti, as the chairperson of IBASC 2021, dear Professor Dr. Samir Kumarkanal, Dr. Yoshiyuki Ueno, and Professor Ashok Mande as the speakers for today's workshop. Welcome to our mini class on aerobic digestion research and scientific writing workshop as part of the International Bioprocessing Association Subject Conference, IBASC 2021. It is an honor for us to welcome all of you. First of all, let me introduce myself to our dearest ladies and gentlemen today. My name is Yosafat Teofilo, and I will be the master of ceremony that will guide today's event. The International Bioprocessing Association Subject Conference 2021 is an event organized by the Department of Chemical Engineering, Faculty of Engineering, Universitas Gadjah Mada, in collaboration with the International Bioprocessing Association. The committee is honored to host today's event, where speakers, experts, students, and researchers in various branches of bioprocess engineering gather. IBASC 2021 will be held for 3 days, from August 3rd to August 5th, 2021. There will be conferences on day 2 and 3 that will cover a broad scope ranging from biomaterial engineering, environmental biotechnology, upstream and downstream bioprocess engineering, bioenergy/biofuel, food technology/engineering, and wastewater resources. We hope that the conferences will be a precious stage for participants to share the knowledge, experience, and the state of the art in the research and technology for bioprocessing application. In order to make sure that this event stays conducive, I would like to remind all participants to mute their mic during the event, unless permitted by our crew, and we hope that you will listen to the whole event eagerly. Before we start, let us pray according to our respective religions and belief. Praying is welcome. Praying is enough. The International Bioprocessing Association Subject Conference 2021 pays close attention to safety in every activity. So, it is our responsibility to read the safety procedure in our event today. Currently, we are in our respective room. Pay attention to the nearest door that can be used in case of emergency. When doing an evacuation, please remain calm, stay alert, and follow health protocols. Before going further to the main event, I would like to read today's workshop agenda. The first one is registration and opening class. The second one is opening by Master of Ceremony. The third one is the opening speech by the Head of Department of Chemical Engineering, Mr. Muslihin Hidayat, PhD. The fourth one is lecture class 1 with anaerobic digestion as the topic, and will be led by Professor Dr. Samir Kumar Kanal. The fifth one is lecture class 2 with microbial ecosystem on thermophilic and aerobic digestion as the topic, and will be led by Dr. Yoshiyuki Ueno. The sixth one is lecture class 3 with scientific writing as the topic, and will be led by Professor Ashok Pandey. And the last one is closing by the Master of Ceremony. As for the start, I would like to welcome Mr. Muslihin Hidayat, PhD, as the Head of Department of Chemical Engineering, to give welcome speeches for today's event. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. My name is Muslihin Hidayat. It is such an honor for me to speak on behalf of the Department of Chemical Engineering, Faculty of Engineering, Universitas Gadjah Mada, Yogyakarta, to extend our warm greeting to welcome you to the International Bioprocess Association Subject Conference, or IBASC 2021. All praise belong to God, the Creator of the universe, and let us to grateful to Him for all blessing, mercies, and graces. All distinguished speaker, Prof. Samir Kumar Kanal from the University of Hawaii at Manoa, Dr. Yoshiyuki Ueno from Alzheimer's Lab Japan, Professor Ashok Pandey from the Center for Innovation and Translational Research, CSIR, in the Indian Institute of Toxicology Research, Lucknow, and Editor-in-Chief of Bioresources Technology. Dr. Ricky Hikmawan Warga Kusuma from Indonesia, Professor Yurina Zainal Abidin from Malaysia, Professor Marasu Tanaka from Japan, Professor Meng Yiwei from Taiwan, the moderators and participants for IBASC 2001 and the participant for WAFER Summer Course 2001. Welcome. It is a great pleasure to welcome you to the International Bioprocess Association Subject Conference IBASC 2021 on sustainable technologies for bioresource utilization, bio-based product, bioenergy, and environmental protection in Yogyakarta. We thank to the International Processing Association, or IBA, for the support as our partner who jointly organized the conference with Universitas Gadjah Mada. We also thank to the conference organizing committee, particularly Dr. Yuni Kusumastuti as the chairperson, whose members have worked so hard to organize this conference. Let me walk you through the series of activities of this year's International Bioprocessing Association Subject Conference. The conference will be held virtually on August the 3rd till the 5th, 2021. They are also called pre-event, as our kick-off on August the 3rd, which consists of mini class on current advanced anaerobic digestion research technology and high impact publication workshop. The speakers of the pre-event are Professor Samir Humar Kanal, Dr. Yoshiyuki Ueno, and Professor Ashok Pandey. The IBASC Conference 2021 are delivered with a Zoom platform which can accommodate 87 papers for all over the world are going to be held in August the 4th till the 5th. The plenary lecture is going to present by Dr. Riki Hikmawan Warga Kusuma with topic Striving for National Chemical Industry Sustainability, the step-by-step green economic approach. And then Professor Yurina Zainal Abidin with a topic Harvesting Sustainable Bioresource for Multivarious Environmental Application. And then Professor Masaru Tanaka with a topic Design of Biocompatible Material for Advanced Medical Devices and Tissues Engineering. And Professor Meng Yiwei from with a topic Surface Modification by Plasma for Application in Biomedical Material and Devices. We do hope that the conference become a precious stage for participants to share the knowledge, experience, and the state of the art in the research and technology for bioprocessing application, as well as a forum which could provide opportunities for further collaboration among the participants. In conjunction with IBASC 2021, we also hold the Indonesian Society for Smart Biomaterial and Tissues Engineering meeting on the last day of the conference, which is the August the 5th. We expect that the meeting provide a platform for researchers from various multidisciplines such as engineering, material science, biology, and medical, as well as all stakeholders, including industrial scientists, clinicals, and regulatory professionals to share and to discuss the cutting edge of biomaterials innovation for promoting human health. We would like to conclude my speech by encouraging all delegates to participate an increasing number in all the activities and discussion through a digital platform for the next 3 days. I wish everyone a successful and fruitful conference. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Samir Kumar Kanal as the speaker. Anaerobic digestion is a sequence of processes by which microorganisms break down biodegradable material in the absence of oxygen. For the second class, the topic is microbial ecosystem on thermophilic and aerobic digestion. The lecture is focusing on microbial ecology and physiology, which are crucial to understand the methanogenic reaction in bioreactors. The topic stated will be discussed by Dr. Yoshiyuki Ueno. The last class will be a workshop class with a topic scientific writing with Professor Ashok Pandey. During the lecture, the participants can ask questions to the speaker by sending the question using the Zoom chat feature. Participants with their question selected will be able to ask the question directly to the speakers during the Q&A session. I would like to welcome Mrs. Wiratni, PhD, as the moderator, who will guide the course of our discussion in our lecture class today. Before moving to the lecture, I would like to present a brief profile of our moderator today. Mrs. Wiratni, PhD, is currently specializing in bioprocess engineering, especially in the area of bioreactor design, biorenewable chemicals and materials production, and wastewater treatment technology. Her scientific reputation includes L'Oreal UNESCO National Fellowship for Women in Science, Schlumberger Faculty for the Future Fellowship, Asian US Science and Technology Fellowship, and Universitas Gadjah Mada Best Faculty Award for Science and Technology. Now, without further ado, Mrs. Wiratni is welcome to take the lead of this session. The time is yours.
Thank you, Theo, for the introduction. I will take over from here. Good afternoon, ladies and gentlemen. I'm very happy to welcome you all in our first lecture in the pre-event of IBASC. Although you can easily Google the name of our next speaker, and you will come out with all of impressive information about him. But take this opportunity to introduce our first speaker, Professor Samir Kumar Kanal. He will share to us the anaerobic digestion processes, especially for wastewater and resource recovery. He is currently the Professor of Environmental Engineering at the University of Hawaii at Manoa, and also the Department of Biology at Hong Kong Baptist University, Hong Kong. Prior to his current position, he has quite global experiences in his academic training. He was a postdoctoral research associate for 2 years and then research assistant professor for 4 years in the Department of Civil Construction and Environmental Engineering at Iowa University, USA. He received his BS with honors in engineering from Malavia National Institute of Technology, Jaipur, India, and then MS in the Environmental Engineering from Asia Institute of Technology in Bangkok, Thailand. So that's a quite international exposure of our speaker. He got his PhD degree in Environmental Engineering from the Hong Kong University of Science and Technology. And then currently he is globally recognized in the field of anaerobic digestion, nanobubble technology, aquaponics, and waste to resources. He has over 125 publications in top rated international journals, 17 book chapters, and 10 books, including 1 best-selling books and 1 textbook to his name. So I think that makes him one of the most productive researcher in this area. He is globally, his Scopus H index right now is 42 with more than 6,600 citations. In addition to his academic publication, his scientific publication, he also received several awards, such as the prestigious Board of Regents Medal for Excellence in Research by the University of Hawaii, Manoa, Elsevier Impactful Research Award, CTAHR Dean's Award for Excellence in Research in the College of Tropical Agriculture and Human Resources in University of Hawaii, and also he was ranked the world's top 2% scientist by Stanford University study. He is also an associate editor of Bioresource Technology and serves on the editorial board and advisory board for 4 other journals. It is a very impressive CV of our speaker, so we are lucky to have him to share the experience today with us. Before I hand over the screen to my best friend, Professor Samir, let me brief the participant about the session management. As mentioned by the MC, we will have about 1 hour lecture from Professor Samir. And then after that, I will open the question and answer session for 30 minutes. A participant who wish to ask questions can type the questions in the chat. You can use English or Bahasa Indonesia if you would like. I would be happy to translate. Or you can raise hand and open cam to ask the question directly after the lecture from Professor Kanal. Samir, can you hear me? Are you ready to share the screen? Hi, Samir. How are you? It is good to see. It is bad that we cannot meet in person like 2 years ago, but hopefully this can be the same as before 2 years ago when you gave the lecture in our university.
That's right. That's right.
Further ado.
Yes, yeah. So
I'll share the screen for you.
Yes, I did it. Thank you so much, Professor Bratney, for your very kind introduction, and very good morning, good afternoon, good evening to everyone. And I hope you can hear me well, and I hope you all are staying safe and healthy in this difficult and challenging time. So today, of course, we were not able to meet each other in person, but we'll try to make it very interactive. And today I'm going to talk about general introduction about anaerobic processes, and then followed by the second lecture on the anaerobic processes for wastewater treatment. You see, so this is, if you want to read more information about anaerobic processes, there are 2 books. The one book completely devoted on anaerobic processes. Second one is bioenergy, but there are 2 chapters that focuses on the anaerobic processes. If we look at the energy situation, about 1.5 billion people, which is about 20% of the population, currently do not have access to electrical power or power grid. And about 3 billion people, which is about 40% of the population, rely on the solid fuels such as firewoods, crop residues, cattle manure, coal for the cooking. And then among those, biomass play a very important role. So about 90% of those energy that people use in the rural area are actually coming from the biomass, which is a very important resources globally for especially developing countries. So from their perspective, it becomes quite important to look at anaerobic digestion. So anaerobic digestion is biological processes which are carried out by the microorganism in absence of oxygen, and it converts the organic matter into methane, which is a gas, and carbon dioxide. Anaerobic processes occurs in nature, including ocean, deep ocean, lake sediment, marshy land, paddy field, and even the digestive tract of human being and animals. So this is a very commonly found processes in nature. So why anaerobic processes? Why anaerobic digestion? Because it converts the waste material into energy using different type of feedstocks that can be used to produce electricity, but can also be converted into transportation fuel that can be used in car and other type of vehicles. And it can handle different type of waste materials, food waste, sewage sludge, biomass, and so forth. The technology is very mature. There are thousands of plants operating globally, producing energy and treating waste. And this process is also very ideal in a location which are very far from the electrical grid. So they are very good for decentralized application. And also anaerobic processes has the potential to cut down, to curtail, or to reduce greenhouse gas emission. And of course, this is one of the most widely used processes for waste management, for waste remediation, especially many of these solid waste. So if you look at the gist of the anaerobic processes, it contains a bioreactor that contains different type of microorganisms. We supply all the waste material, including manure, domestic waste, or sewage sludge, and then different type of lignocellulosic biomass in the reactor. They stay there for maybe 20 to 50 days, and the microbes will degrade it, producing biogas. And the biogas, we can clean it, remove moisture, hydrogen sulfide, and ammonia, and so forth. And we can use this biogas as a direct burning for cooking, lighting, and heating, or we can actually use the biogas in combined heat and power, producing electrical energy and thermal energy for heating, or we can remove carbon dioxide and other impurities, we upgrade them, and we can inject into the natural gas grid or use for transportation. So these are essentially the energy products coming from the anaerobic processes. The digested liquid part or digested part is called the digestate, which contains solid and liquid. We can store them for some time, and then we can either transport them, apply to the land for agriculture application, or we can separate solid and liquid. And solid can go into composting and be used as a compost, or we can directly apply to the land. So by this way, this provides a kind of a biorefinery where you can, everything is utilized. And for a small scale, it's very commonly used in many developing countries. We call household digester. These are a small reactor, maybe 1 to 3 cubic meter or 1,000 liter capacity reactor. We supply residual food waste or different type of animal waste into the reactor, stay there 20 to 30 days, and produce biogas. But at the same time, most important component is the digestate liquid part or the semi-solid part. These slurry are very rich in nutrient. So what they do, they essentially apply this one into the rice field to produce rice. And then because it has a different type of nutrient, nitrogen, phosphorus, potassium, micronutrient, they also use for horticultural application, means they can use for growing fruits and vegetables. You can see here, there is bitter melon, different types of herbs. They use actually this digestate. But they also sometimes apply it to the fish pond, and they actually grow fish. So they're essentially converting this waste into different type of food and feed product for human beings. So this way, they provide a lot of income for those rural areas, rural communities. So therefore, anaerobic digestion in a small scale also provides a lot of economic benefit to the rural communities. Now, anaerobic digestion is not a new technology. It has been there for centuries. If you look at the history, about 10th century BC, Assyrian, they use this biogas for heating the bath water. They also use in 16th century, Persian use this biogas for also heating the water. So therefore, they are quite old technology. They have been there. But scientific discovery of the biogas or methane was actually done by the Italian physicist Alessandro Volta when he was on the summer holiday in Lake Maggiore. He found out there's a burning gas, and he referred this as a methane gas. And he was also the first one to relate the amount of gas produced depending on the organic matter. So therefore, he was the one who actually did some scientific explanation of the anaerobic processes and methane gas. Now, in anaerobic digestion, the substrate, or we call feedstock, are essentially written down as carbon, oxygen, hydrogen, nitrogen, and sulfur. So this is the organic matter. And sometimes we add some nutrient, different types of nutrient, could be cobalt, nickel, molybdenum, and so forth, iron. Anaerobic microorganisms degrade the organic matter, converting into methane, CO2, and produce ammonia and new cells. So these are essentially the main product we are looking at biogas, mostly methane and CO2. Anaerobic processes can be divided into two parts. One is called anaerobic fermentation. Second one is called anaerobic respiration. And these two processes takes place simultaneously in the anaerobic digester. So slightly more explanation. In anaerobic fermentation, there is no external electron acceptor. So that means the electron produced during the degradation of organic matter are accepted by the organic matter itself. So organic matter itself will act as electron donor and electron acceptor. And therefore, in these processes, it produces a less amount of energy, and most of the energy are remain into the fermentation product, including methane, ethanol, butanol, hydrogen. So if you take a very simple substrate or waste, if you have glucose here, during the anaerobic fermentation, this is broken down into pyruvate, which is a two-carbon compound producing energy and electron. And then same electron is actually taken up by the organic matter itself and producing ethanol. So you don't need any electron acceptor. Organic matter itself will act as electron acceptor, and all the energy are conserved in the ethanol. So this is the fermentation processes. So anaerobic fermentation is really important to produce bioenergy and biofuel. If you look at the process in anaerobic digestion, we produce methane. So about 2/3 of the methane is produced by anaerobic fermentation, such as here, acetate converting into methane is by anaerobic fermentation because you don't need any external electron acceptor. We produce ethanol from different type of feedstocks, starch or glucose, also by anaerobic fermentation. Butanol also produced by anaerobic fermentation. Hydrogen is also produced by anaerobic fermentation. So all the energy are actually conserved in these products. But in anaerobic respiration, we need external electron acceptor. The electron acceptor could be sulfate, nitrate, or carbon dioxide. And during these processes, more energy are actually produced. So if you look at the example of glucose here, when the glucose is broken down in the anaerobic respiration, it produces pyruvate and produces both energy and electron. Now we have here external electron acceptor. It could be sulfate, carbon dioxide, and nitrate. So what they do, they essentially comes and acts as an electron acceptor, takes the electron produced by the breakdown of glucose and converting sulfate into sulfide, CO2 into methane, and nitrate into nitrogen. So this converting nitrate into nitrogen is called denitrification that takes place under anoxic conditions. And at the same time, then the pyruvate are broken down into CO2 and water. So in this processes, you can see when they use electron acceptor, CO2 as electron acceptor, they produce methane. If there is sulfate, they produce hydrogen sulfide. So this is the process of respiration. So in the biological system, microbes always prefer oxygen. So oxygen is the first preference for the microbes to take as an electron acceptor. If there is no oxygen, especially in the anaerobic processes, they go for the next electron acceptor, which is nitrate. If there's no nitrate, they go for sulfate. And CO2 is the last or least preferred electron acceptor because they will get less amount of energy by taking this as electron acceptor. So they always prefer electron acceptor that provides them the much higher free energy. And that comes from the oxygen as an electron acceptor. But in anaerobic processes, we don't have oxygen. So electron acceptor could be nitrate, sulfate, or CO2. So only when you don't have nitrate and sulfate, you produce methane by using CO2 as an electron acceptor. So how can we estimate how much methane you can produce from the substrate? So there are three different approaches. There are three different ways. First number is when you know the chemical structure of the compound. For example, I'm going to tell you a little bit more later about this one. And in some cases, we really don't know what is the chemical structure of the compound, but we know how much organic matter that compound has, which we call chemical oxygen demand COD. And last, we don't know chemical structure. We also don't know what is COD, especially the waste with a very high solid content, such as food waste, sewage sludge, and a very complex waste. And these are the third way. So three different ways of calculating or estimating how much methane you can produce from the organic matter. So we're going to look at the first one, when we know chemical structure. This chemical structure can be determined in the lab by proximate analysis. You will know how much carbon that compound has, how much hydrogen it has, how much oxygen, nitrogen, and sulfur that compound contains. Based on this proximate analysis, you know the chemical structure, and you can calculate how much methane you can produce. So here, this is the example. It has carbon, hydrogen, oxygen, nitrogen, and sulfur as the organic compound, and then different amount of atom it has. It has A atom of carbon, B of hydrogen, C of oxygen, and so forth. This you can find out by proximate analysis. And by using an equation called Bushwell equation, you can theoretically convert this one into methane, CO2, ammonia, hydrogen sulfide, and then some cell. So these processes will tell you, based on the chemical structure, maximum how much methane you can produce. And this is the stoichiometry. And then if you solve their function A, B, C, D, E, this is known to you. So unknown are here. F is unknown, which is a coefficient. G is unknown coefficient. S is unknown coefficient. I is unknown coefficient, and J is unknown coefficient. There are 5 unknown. You can derive 5 equations. You solve the equation, you can get the coefficient in terms of known A, B, C, D, E. So this equation will give you the coefficient value, and this coefficient will give you how much methane you can produce by the complete degradation of the substrate. And for different substrates such as cellulose, starch, hemicellulose, protein, lipid, primary sludge, and secondary sludge, we can find out their composition by proximate analysis. And based on this known composition, we can estimate how much methane you can produce, and so forth. So I'm going to give you one example here. I'm taking example of glucose, which is C6H12O6. So A is 6, B is 12, and C is 6. There's no nitrogen, so N is 0, no sulfur, S is 0. So, and sorry, here, so D is 0, E is 0. So you can substitute this value in this equation. And you can also use this equation to calculate how much methane you can produce. This is the coefficient for methane, which is this one. And the denominator part is this part, A, B, C, D, E. You have to add all. Carbon has 12 atoms, so 12A. Hydrogen has 1 atom, so 1B. Oxygen has 16 atoms, so 16C. Nitrogen has 14 atoms, so 14D. And sulfur has a 32 atoms, so 32E. So you can solve this one, and you can find out how much methane you can produce from 1 kg of glucose. And you can actually find out that 0.373 cubic meter of methane can be produced from 1 kg of glucose under STP. STP stands for standard temperature and pressure. That means 0 degrees centigrade temperature and 1 atmospheric pressure. So by this way, you can calculate maximum how much methane can be produced from glucose. You can do for some sucrose, fructose, biomass. You can calculate by using this equation. But your temperature is different. Say temperature is 35, then you have to make temperature correction, and you can find out also the different temperature. This is the simple way of calculating the methane yield. If you don't know chemical composition, you know how much organic matter the waste has. So different type of food processing wastewater coming from different industry. You can measure how much chemical oxygen demand that has. We measure this in terms of COD. Then you can also calculate the amount of methane you produce from this type of waste stream. So there is a simple example, how much methane can be produced for complete anaerobic digestion of 1 kg COD. COD you can measure in the lab by using different methods. There are methods in the standard books, and you can actually use these traditional methods to find out the COD of the organic, the waste. So what you do at the first step, you convert this methane into the COD. So if you completely oxidize methane using oxygen, you get CO2 and water. And finding out the COD of methane based on the molecular weight. So methane is 16 grams, oxygen 32 times 2 is 64. So 1 gram methane has a COD of 4 grams. Now you have found out the weight. You have to convert this methane weight into the volume. So you use gas law. According to gas law, 1 mole of any gas at standard temperature and pressure will occupy 22.4 liter of volume. So you can convert this weight into volume by using gas law. So 1 mole methane means 22.4 liter. 1 mole means 16 grams. So 1 gram methane has a volume of 1.4 liter methane. So by combining equation 1 and 2, you will find out that 1 gram methane is 4 gram COD, which is 1.4 liter methane. Then you can find out from this one, 1 kg COD can produce 0.35 cubic meter methane. So under standard temperature and pressure, you see that 1 kg COD will produce 0.35 cubic meter of methane. So that means by knowing the organic matter, which you can measure in the lab, you can estimate how much methane you can produce from this given wastewater. Okay? So this is also another way of calculating the theoretical methane production. But this is for STP, but if you want to convert it into higher temperature, 35, you can use temperature correction again and find out the methane yield at 35 degrees centigrade temperature. Just one example, suppose you are working in an industry and you want to know if you have a wastewater, how much biogas you can produce. You can also calculate by knowing the chemical oxygen demand, by knowing the COD. So you have to measure COD of wastewater, which you can measure in the lab. And suppose the COD of the waste is 40 gram COD per liter, and you produce 10 cubic meter of the wastewater every day, then you can find out how much methane you can produce. So you convert this into COD load. Load means flow rate time concentration. So you can multiply this, have to be the same unit. So 10 cubic meter, and this is 40 gram, which means 40 kg per cubic meter. So you'll see you're going to produce about 400 kg COD per day from the wastewater. And based on our calculation, 1 kg COD will produce 0.35 cubic meter of methane at STP. If you have 400 kg COD, you will produce 140 cubic meter COD per day at STP. If you want to convert in 35, you do temperature correction. You have to convert this temperature into Kelvin, okay, not centigrade, because that's the standard temperature. So you convert this one into Kelvin, that means adding 273, you get 158 cubic meter methane per day by this wastewater. So by knowing this calculation, you can estimate whether you can produce enough energy from your wastewater, and you can make some design, some calculation in the cost analysis part. So that's another way of calculating methane production. Now, methane has the energy value. So if you look at the energy value per unit volume, 1 cubic meter of methane contains about 35,846 kilojoule at STP. And in English unit, this is about 1,066 BTU, British thermal unit per cubic feet. So usually we say 1,000 BTU, 1 cubic feet. In terms of the mass, the heating value of methane is about 28,850 BTU per pound mass, or 55.4 megajoule per kg. And you can see that if you look at the coal that we use in the power plant, that has energy value of 30.1 megajoule per kg. So you see biogas in terms of mass has almost 2 times higher energy value. So they can provide you energy. And if you look at the biogas, there are some waste, food waste, different type of slaughterhouse waste, very complex waste. You don't know how much is your COD, you may not know your composition. Then we use different way of calculating the biogas. So we usually do what we call the lab scale BMP test, biomethane potential test, or pilot scale test. And we conduct this study for some time and calculate the methane yield. So in the lab, we do BMP test. We put that high solid substrate. We put inoculum. Inoculum is microorganism. And then we run them for maybe 30 or 40 days and find out this curve. This is the methane curve production, cumulative methane production per unit time, and look at the maximum methane, how much you produce. So this is one way of estimating how much methane you can produce from those type of high solid waste. And different high solid waste has a different composition. Maize silage, which is corn waste, green corn, green cutting, which is essentially some of the grasses, straw, cattle manure. So they have a different yield. This is essentially the per ton of the this is essentially the metric ton. So 150 normal cubic meter per metric ton from maize silage. So you can calculate how much methane you can produce per ton of your waste material. So this is one way of calculating your methane yield. So what are some of the benefits of energy anaerobic digestion? Of course, it produces energy that could be used for fuel application, could be methane or hydrogen. It is also a way of producing, treating the waste. It can actually reduce the waste volume, waste mass. Also, it reduces the biodegradability of substrate. The substrate can be degraded, so it become more stable product that can be then applied or as a fertilizer. It doesn't produce any type of odor, any type of nuisance. So it reduces odor production, emission of greenhouse because you capture everything. And also it can reduce pathogens in your waste, such as E. coli, Salmonella, and Shigella, those kind of pathogens. And we study also found that anaerobic digestion can actually reduce your many type of antibiotic resistant genes in the bacteria, and also the weed seeds. Many of the animals, they go and they when they eat their food, they contain different type of seeds, grass, weeds. And then when they digest it, for anaerobic digestion, they reduce the germination of the seed. So it actually, when you then apply, you don't see any type of those weeds coming in the field. So there are some of the benefits of anaerobic digestion. Now we talk about general overview, but how the anaerobic processes work in the feed or in the digester. So this is a biological processes. Anaerobic, if you look at the substrate, it's very complex. It contains protein, different protein, carbohydrate, and lipids. So we divide it into three major components in the substrate. So in the first step is called hydrolysis, which is also liquefaction. You break down this complex waste material into simple soluble product. Protein goes into amino acids or peptide. Carbohydrate would go into soluble sugars, could be glucose, fructose, sucrose, so forth. And lipid will be broken down. This is fatty product, broken down fatty acid and alcohol. So this is the first step for hydrolysis, which means breaking down into soluble product. And this is done by the fermentative microorganisms. Then after that, the soluble product that we produce by the hydrolysis are broken down into intermediate products, which are essentially called the volatile fatty acids, which has the carbon compound more than 2, such as propionate, butyrate, valeric, caprate. It also produces lactic acid or lactate and ethanol. So these are the degradation intermediate products produced from this product. This is the process called acidogenesis because it produces acids from this product. It can also produce actually some acetic acid directly, okay, from this product. And also it can produce hydrogen and CO2 directly from this product as well. Now, after this intermediate product, what happened there? There is a second step, third step.
Recording in progress.
Within the system. And last step is converting this acetate and hydrogen and carbon dioxide into methane. So these are the two different processes we call methanogenesis. In one step is called acetoclastic or acetotropic methanogenesis, where acetate is converted into methane and carbon dioxide. Second is called hydrogenotropic methanogenesis, where hydrogen and carbon dioxide are combined, converting into methane. So major part of methane is coming from acetoplastic. About 28% is coming from the hydrotropic methanogenesis for sewage sludge. It may vary slightly, but this is the major part. So this is the pathway how the complex organic matter are broken down and converted into methane. Now there are limiting steps. If your waste is very high in solid concentration, food waste, okay, sewage sludge, biomass, in this case, your hydrolysis become your rate limiting steps. Okay, this step becomes rate limiting steps because the microbes will not be able to degrade easily. So we sometimes do some kind of pre-treatment to enhance the hydrolysis. And if the waste is highly soluble, such as glucose, fructose, sucrose, so forth, then your rate limiting step is a methanogenesis. Okay, last step. For soluble wastewater, methanogenesis become the rate limiting step because they produce too much acetic acid that the microbes